really my pleasure to introduce Dr. Ivan Liachko. So Ivan earned his bachelor's degree in biology from Brandeis University, and then his PhD from Cornell University in genetics and development. He then did a postdoc at Cornell and Washington State University. And for over 2 decades, he studied the molecular genetics and epigenetics in model and non-model organisms. And he has extensive experience with molecular biology of DNA and chromatin, as well as computational methods. Over the last 10 years, he has specialized in developing novel molecular tools that employ massively parallel sequencing technologies to elucidate genomic mechanisms. He adapted Hi-C towards scaffolding and assembly of diverse genomes and metagenomes. And then he went on to found Genomics to commercialize this technology for genome assembly and diagnostic applications. So without further ado, I will let Dr. Liachko tell us a little bit more about connecting the Virome, Plasmidome, and Resistome to the microbiome using Proximity Ligation Technologies. Welcome, Ivan.
Thank you. Thanks, Yana. Thank you guys for coming. Thanks for having me. It's really fun. I haven't, I don't do too many academic seminars these days, especially after COVID, like traveling around and whatnot. So it's a real pleasure. What I'll do is, so I'm here representing, a small company that we started in Seattle called Phase Genomics. Phase is a spin out from the University of Washington. And so, kind of part of this talk is going to be, I'm going to kind of talk a little bit about sort of the process of how it all came about. So not so much about founding stuff. You can talk to me about the company stuff later if you want. But really this talk, I have this kind of silly title, but it's really intended to be sort of about, the theme is really about information. And so the talk is really about capturing information in kind of new and creative ways, and then like learning to do new things with it. And I sort of will show you a trick that we developed, sort of exploited, that was originally developed for something, and we've repurposed it for something else. But the theme is really about information and capturing novelty. So I think most of you guys are genomics people or know a little bit about genomics. I'm going to give you a 1-slide summary of genomics. Kind of goes like this, right? So we want to sequence everything in the world. But the problem, of course, we run into is the reality of what that looks like. And right, you're trying to capture these really sort of, you're trying to capture genomic information in these genomes, but you have to go through this gauntlet of starting with very short reads, right? Now, again, the short reads carry some information in them, but they're also missing lots of information. And you try to assemble them, right? Usually you assemble reads into contigs, and then you try to assemble contigs into some form of a genome, trying to sort of recover as much information as you can that was lost. And this is hard enough when you're dealing with a single genome, like a plant or an animal or a fungus or something, right? It's even harder when you're dealing with metagenomes, with microbiome samples, where you're simultaneously dealing with lots and lots of organisms. And so what we've done is we took a trick that was developed for a particular application and utilized it for a number of different directions. What I'll do is I'll back up a little bit and I'll tell you about the underlying technology that we're going to use and what it's used for. And I'm going to first talk about like genome assembly and some human like cancer stuff briefly, just to give you some context. And then I'll talk about microbiome stuff for most of the talk. So the underlying principle behind what we're going to talk about today is that we think of genomes like we draw them as like linear chromosomes or maybe these little butterfly things, right? But these are actually just mitotic chromosomes, right? So normally this is what your genome actually looks like, right? It looks like a big ball of spaghetti, right, inside of a cell. And within this three-dimensional structure, there's lots of interesting biology that goes on. And so people have developed tools to try to understand how a genome folds in 3 dimensions. And so these tools are typically called proximity ligation. So what they essentially try to do is you take cells that are still intact and you cross-link them. So, right, so when you still have the spaghetti ball, and you treat it with formaldehyde, usually it's a cross-linker, and it goes inside the cells, and it cross-links DNA, protein, RNA together. And when it does so, DNA molecules that are close together get linked, get basically physically trapped next to each other. You can then basically chop up this cross-linked chromatin, you liberate these little DNA ends, and you ligate them together, and you isolate these chimeric junctions. So, right, so when you do this, this is called proximity ligation. It's when you imagine, if you guys have ever tried cloning a plasmid, right? You know, you're trying to ligate two pieces of DNA together, but they don't want to ligate to each other. They want to ligate to themselves, right? They want to close into little circles. And that's really because those ends of that DNA are physically kind of linked. They're like next to each other, right? They're constrained. And so the same thing here. So when you have sequences that are physically stuck together, they're going to preferentially ligate to each other, and they're going to create these little chimeric junctions, right? Sequences like this red-blue sequence. You're going to get a ligation between two molecules that may be on very far away parts of the genome, but they're physically close in three-dimensional space, right? And then through a number of other steps, we isolate these junctions and sequence them. When you do that, that tells you that sequence red and sequence blue were physically close together in three-dimensional space, right? And what you do is essentially, because you're doing this with sequencing, with next-gen sequencing, you're doing this millions and millions of times, right? You're essentially counting how often everything is touching everything else in the genome, right? You're counting how often every part of the genome is touching every other part of the genome, and then you extrapolate them into these matrices, and you can extract a 3D model for the genome that way. And so this technology, this proximity ligation tech, was originally intended to do this, to try to basically, again, remember, we're talking about kind of capturing information. This is capturing a funky kind of information. It's capturing how often sequences are touching each other in 3D, right? Which is like a little bit of an unusual thing to look for when you're doing sequencing. But when you do this, you capture other kinds of information that the original inventors did not intend. And we decided that that information is actually even more useful than this 3D stuff. And so the thing that I'll talk about first a little bit is that when you're capturing, when you're measuring how close things are in 3D, you're actually also measuring how close they are in 2D. Okay, because imagine a piece of spaghetti and it's floating around. Sequences that are close together on that spaghetti are more likely to occupy the same space than if they're on opposite ends of it. Then they're less likely to find each other, right? And it's not even really biology, it's just physics, right? Just it's polymer dynamics. So when we're measuring how close, basically I guess what I'm trying to say is, as sequences get closer together, they touch more, and if they're further apart, they touch less. Right. And so it's kind of like a genetic map, right? Like when you're trying to map a genome, back in the day, they used to do these recombination maps with centimorgans and that kind of thing. You were trying to figure out how far and how close every part of the genome is to reconstruct it. And here we have the same kind of thing with the sequencing data. We're essentially measuring which parts of the genome are far and which parts of the genome are close. And so what that means is, if you have a genome and you don't know how to assemble it, you now have information that tells you which sequences are far away, which sequences are close. And so if you have a genome, and let's say for like a plant or an animal, it's never been assembled, you have pieces. What this data will do is it'll tell you how to put it together, because it'll tell you, like, okay, you have these pieces. Sequences that are in the same chromosome will touch more than sequences between different chromosomes. Sequences that are close will touch more than sequences that are far. And so this gives you the information you need to basically what we do, it's called scaffolding a genome, where you have little pieces, these contigs of a genome, and you're trying to put them into chromosomes. And so we sort of, we kind of showed that you can do this with Hi-C data. We've also shown that you can phase a genome. So for those of you who don't know what phasing is when you have, let's say you have a diploid organism, right? You have, like you guys all have diploid genomes, right? You have one chromosome from your mom, one chromosome from your dad, and they have some unique mutations on them, unique SNPs, right? Well, because your mom chromosome and your dad chromosome are physically two separate objects, the sequences on your mom chromosome will be touching other sequences on your mom chromosome, and the sequences on your dad chromosome will be touching other dad sequences. So you can not only use this information to scaffold the genome, you can also use it to separate apart the haplotypes. You can figure out which mutations are on sort of your mom haplotype and which mutations are in your dad haplotype. So we did some work on this. We published some stuff with together with PacBio to basically create what's called platinum genomes. So a genome that's not only in chromosomes, but also it's phased. And again, this is just, we're using three-dimensional genome data to do something very not three-dimensional, right? To actually figure out how to separate haplotypes from each other and also how to put the chromosomes together. And so I won't talk about this because it's sort of more for dealing with macrobes. Who cares about those guys? We want microbes. But this is something that we've published a lot on now. We're getting close to like 200 papers on the subject. I have a few, because of my job, yes.
Yeah, I'm a little confused if you can just sort this out before you get too far in. Are you still doing the cross-linking in vivo or are you doing it on naked DNA?
Right, so the question is, are we doing the cross-linking in vivo? And the answer is yes, and that is the key here, is we're cross-linking before the cells are broken, before the DNA is extracted. So kind of one way to say, like, how do you assemble, how do you scaffold an entire chromosome? Well, don't break it in the first place, right? So we're trapping them. The power of this technology lies in the fact that we're trapping this information before the DNA is extracted. So before we don't have to deal with high molecular weight DNA extractions. We don't have to deal with breaking the DNA. The entire chromosome is captured. And so it's basically capturing interactions at infinite distance, right? Doesn't matter how big the chromosome is, it jumps any distance apart.
So I guess my point of confusion is that aren't there active systems to keep DNA in proximity inside the chromatin domains and stuff like that?
Yes.
Whereas your
right, so
your simple proximity along the chromosome is going to be more dependent on random interactions.
Yeah, the question is, what about the 3D genome folding, which is what this technology was originally designed to look at? Doesn't that interfere with this scaffolding? So here's a heat map. So what you're seeing on this heat map is, this is basically how this IC data is plotted. X coordinates and the x-axis and the y-axis are the same. And you're essentially looking for how much interaction there is between every pairwise combination of coordinates, right? And what you see here is all this stuff out here in the distance. This is your three-dimensional signal. These are your loops. These are your topologically associated domain, your chromatin domain, etc. This big diagonal in the middle, right? That's your proximity signal, right? So the principle here is that yes, you do get three-dimensional signal. But the proximity signal is orders of magnitude stronger. In fact, the way we came up to this is that when people did three-dimensional modeling of a genome, they would want to, they're using this stuff and they need to throw this away because it's so strong, it swamps all the data. So in general, the proximity signal was so strong that if you didn't remove it, it would basically nullify all your three-dimensional studies. So people were treating this as junk and throwing it away. And for us, it's the other way around. This is the strong stuff. This is what tells you how to put your genome together. And as you can see, it's vastly larger. So the idea is that yes, that exists, but the physics beats biology. And so the three-dimensional loops and things, they exist, but just how close, how far stuff is like orders of magnitude stronger.
Got it, thanks.
And so, yeah, and the same thing with phasing, right? Because it just tells you, is it the same chromosome or not same chromosome? Like that signal is going to be way stronger than like a loop that may exist in some cells and not in the others, right? Because the phase is the same in every cell, more or less, right? So it's something we'll be publishing a lot on. Another thing that we can do with this, and this is another, it's related to the previous question. So let's say you have two samples. These are two human samples, and it's the same this heat map. So you're looking at how much proximity interaction there is between sequences in this box, like chromosome 2 and chromosome 2, chromosome 3 and chromosome 3. So the heat inside of these boxes means that different parts of chromosome 2 are touching other parts of chromosome 2, but they're not touching other chromosomes quite as much. But then if you look at this sample, which is a cancer sample because they labeled it here, there is this hot spot here, right? What that means is that this part of chromosome 2 is touching this part of chromosome 4 a lot more than it's supposed to. Why could that be? Because there was a translocation in it. Right. So if you can scaffold the genome de novo without knowing how any of the pieces belong together, if you have a reference genome and there is a structural variant in your sample, you can see them right away. right? So this turns out to not only be a useful tool for genome assembly, it's also a really good tool for basically reconstructing karyotypes, right? So this is something that's applicable to cancer people. I don't know if there's any cancer people in the audience, right? Normally what you do is you do assays like this. So if you have, like, for example, if you got leukemia, right, they would do this thing called karyotyping, where they would actually look at your chromosomes under a microscope to try to detect rearrangements, right? This is super commonly used diagnostic. And then there's this thing called FISH, fluorescence in situ hybridization, where they do the same thing. Basically, chromosomal anomalies, chromosomal rearrangements are oftentimes drivers of cancer. And so they look at this all the time. What's interesting is this data is actually really good for seeing that, because if you can see these sort of hotspots in the maps, you can immediately detect translocation. So these are all the things that I won't focus on. I just wanted to kind of get you guys started, kind of thinking about, like, we have this assay for looking at how a genome folds in 3 dimensions, but we can use that information for all these other things. We can phase and scaffold like a hexaploid wheat genome, right? Things that were impossible before. We can do like diploid genomes, we can do platinum genomes. We can replace these like ancient diagnostics, right? That are used all the time. One other cool thing about this, I don't know if you guys are cancer aficionados, but 95% of all cancer samples are stored in this format called FFPE, which is formalin fixed, paraffin embedded. Like almost every solid tumor sample is stored in FFPE. You can't do cytogenetics in FFPE. Like this stuff here, you do it in FFPE because the sample is all fragmented, the cells are all dead, right? But our method works on FFPE because, remember, formalin is the first step in our process. So this is the other kind of power of this, is suddenly this vast amount of cancer samples could never be assayed for chromosome structure before, and suddenly we can do it with the sequencing trick. And it's not a brute force approach. It's just a library and some sequencing. The trick is that we're extracting this unique kind of information. So you can do all this stuff in genome reconstruction with this data. I haven't talked at all about the 3D genome stuff, right? It's all about like reconstructing genomes. What about metagenomics? So as you guys know, when I talk about like genome assembly, it's like assembling like the world's worst jigsaw puzzle, right? Where like 80% of it is like the sky, it's all blue, you don't know where the pieces go, there's all these duplicated pieces and stuff like that. With metagenome, it's even worse, because now you have like, it's like having like, I don't know, 100, 1,000 jigsaw puzzles. You dump them all together, you mix the pieces. Now you don't even know which pieces go where, right? So you now have, you're sequencing DNA from multiple organisms at the same time. It all kind of becomes this metagenome alphabet soup. You don't know which sequences belong to which organisms. You try to figure out ways to bin them, to group them. If you have a mobile element like a plasmid or a virus, like a phage, right? Once you've extracted the DNA, like you don't know where that plasmid came from, right? Because it's liberated, it's not part of the genome. And the same thing, there's all sorts of other problems. Like if you have genomes that have more than one chromosome, how do you know which chromosomes belong to the same organism? That kind of stuff. And so because of this, there's like, most metagenomic data is really underutilized because we sequence the stuff. But we've lost all this contiguity information because we extracted DNA. So here comes this proximity ligation method. So it works, right? We take samples, we cross-link them, and then we create these little chimeric junctions and sequence the junctions. These junctions tell you that the 2 sequences were near each other in 3-dimensional space. But because the cross-link happened before you broke the cells, right? All these little junctions must have been formed inside the cells. And so what happens, so the idea is that, right, anytime 2 sequences interact by this assay, they must have been inside the same cell at the start of the experiment. Because you made the little junctions, the trap little connections before the cells were lysed. So now, if you have a metagenome like this, right, suddenly you have connections both intra and interchromosomally, right? And now you have your alphabet soup metagenome. You can say this contig and this contig and this contig, they were touching each other. They must have been part of one organism. These were touching each other. They must have been part of another. So you can separate. It's like when you have 100 jigsaw puzzles, but what if you like spray painted them all different color before you mix them, right? Suddenly you know which ones came out of the same puzzles. And importantly, this is my favorite part, is that you can actually, if you have a plasmid or if you have a phage, it will connect to the genome of the host. So you can now, without culturing from sequence data itself, you can figure out which mobile elements are coming from where. And so this is what I'm going to talk about in sort of the rest of the talk. The first experiment we ever did is we tried this on a mixed community. So I come from a fungal background, so we had a bunch of yeast species that we were working with. So we mixed a bunch of yeasts together and did kind of we sequenced them with shotgun sequencing, and we did this proximity ligation stuff on it. And what you're looking at here, the soccer ball of science, is every dot, every node in this chart represents a contig from a metagenome. And the lines, the edges that are connecting them all, that's your Hi-C signal. That's your proximity ligation signal. And so if this was just a regular metagenome, it would just be a gray field of dots, disconnected dots, right? But you can see how this proximity signal grabs the contigs and pulls them into these clusters. Okay. And so we say, well, what are these clusters? What are these like different color blobs? Well, it turns out they are the 12 organisms that we put into the sample, right? And so this was the first ever proof of concept we did. It was all organisms for which we have genomes already. But one key thing is they're all eukaryotes. So they have multiple chromosomes, right? So some of them have 16 chromosomes, some of them have 10, etc. They have plasmids, they have like other stuff in them. And so this showed us very early on, I mean, again, this is almost 10 years ago now, that we could deconvolve even eukaryotic communities, right, without any culturing this way. And then we started doing, and by the way, if you want to go further, you can actually go one step further, because we have Hi-C data for every one of these organisms, right? When you have Hi-C data for the mix, you have Hi-C data for all the organisms in the mix at the same time. So you can simultaneously actually reconstruct those genomes into chromosomes, like I said before, right? So you can simultaneously deconvolve the communities and scaffold those genomes into chromosomes at the same time. So you can get basically plasmid grade genomes right out of a mix. We then started doing lots of different, here's our first ever real metagenome. So we went into like a hipster brewery, we got some open fermentation beer, right? And this is one of these examples where like, we did this, we were gonna do it. I told my PI, I said, I was a postdoc at the time. I said, look, we're gonna go to this brewery and we're gonna get some samples and we're gonna eat their food and stuff and we're gonna charge it to the lab. She was like, can I come? And so, but what we did was, this was an open ferment beer. Again, like if you're not familiar with open fermentation beer, imagine that you have some contaminated culture and you like drink it. Like that's basically, right? That's what it is. They put it, they got some contaminated, it was called Old Warehouse because they like just left this thing open in their old warehouse and then it got fuzzy and then they poured it in some wine barrels and started pitching it. Anyway, the point is we processed it, similar ideas before. And, it wasn't like a complex community. It was only 8 organisms, 4 bacteria, 4 yeasts. But what was interesting about it, and the reason why I highlight it is this green one, this labeled P. membranifasciens, this organism had an interesting property. It had, its genome looked like twice the size of a normal yeast genome. And half of it was mapping to a known organism and half of it was mapping to nothing. And so we thought this was like some kind of crazy software aberration, but then we cultured it, right? This is like beer, so we can culture everything, it's cheap. We cultured it, and it turns out it grows in these like cute little fuzzy colonies. It turns out it's a hybrid. So yeast do this all the time, especially in brewing, where 2 haploids from different species will mate, and they will create this like hybrid diploid, and that will propagate mitotically over time. And that's like how all your lager beers are made. Like there's a hybrid yeast called Carlsbergensis that was the original lager yeast. But the reason I highlight this is that how would you know from a metagenome that there is a hybrid organism? Like let's say you sequenced a metagenome, and how would you know from that data that you have an organism that has 2 genomes inside the same cell, right? Versus just having 2 separate organisms. Right. So this was also the first time we saw that we were able to actually detect hybridization events directly in a metagenome sample without any culturing. So you can find these kind of complex communities. We also learned something very important. If you guys are ever interested in doing easy science that gets a lot of press attention, you should work on beer, because that was a very simple experiment that made it in like all the news and sorts of other things. My like little cell phone pic of yeast is in like the Russian news. So, but anyway, these are sort of like some little kind of proof of concept experiments. But over time, we've been publishing on this quite a lot. We've basically shown that by using this data, so Proximeta is our platform, having this kind of data enables you to do really unprecedented things in the microbiome space. First of all, it just by nature outperforms traditional metagenomic binning, because you now have this added amount of information. In addition to like your normal metagenomic binning that you're doing, you now also know like which sequences are physically touching each other. And so you layer that on top and you can deconvolve metagenomes. So it's a really good way of discovering new organisms that you can't discover by normal methods. It also does something interesting. This may be a little bit of a nuanced point. This is a paper we published recently with Niranjan Nagarajan's lab. He had a lot of human fecal samples that were assembled with long reads, so long read nanopore assemblies and our proximity ligation data. And what you're looking at here is this is the same assembly that's either sort of binned normally, like by normal computational ways, or using our Hi-C data. And so you have the same assembly binned in two different ways. The outcome gives you roughly the same number of bins, and the bins, the mags, are also roughly the same size. So same assembly, binned by two different methods. The number of bins that comes out is about the same. The size of the bins that comes out about the same. But then this figure here, I don't know, you have to read all this. Basically, the purple and yellow, these are high quality genomes. So the number of high quality genomes actually doubles. So what does that mean? When you have same assembly, two different binning solutions, the number of genomes is about the same, the size of the genomes is about the same, but one has twice as many high quality genomes than the other. What that tells you is that normal binning makes mistakes that you can't see, right? Because you're getting the same number of genomes and they're the same size, but one is way lower in quality and it's the same starting assembly. So what that means is that traditional binning makes mistakes by switching contigs around. You can't detect that. And this data allows you to clean that up. And so my point with this slide is just to say that you're not just getting more of them. You're not just getting more high quality genomes. You're actually cleaning up the gunk that's left after sort of normal metagenomes. So it's a really good way of getting into the dark matter of the microbiome. And because you're getting all these, genomes, it empowers your ability to extract more biochemical information. So you process a metagenome sample. If you have more genomes, better genomes, you're going to get better metabolic pathways. You're going to get better metabolic modules. So that's just shown here. This is a given sample. Like this is, I think, a wastewater sample. Basically, yellow boxes are metabolic modules that are, for this sample, only found in like the proximity ligation bin data set and not in the normal data set. And so more genomes, cleaner genomes, more metabolic functions that come out. But one of the things that I mentioned previously, and this is going to be a big part of the talk, is that we can do something else, something that you just can't do with regular metagenomes. And that is the fact that in addition to reconstructing all these little genomes, we're capturing the information that tells you which mobile elements like plasmids and viruses are inside of which cells, right? Because the plasmid will connect to the host because it's physically inside the cell. And so you get linkages between genomes and phages, genomes and plasmids, right? And so you can actually do this without any culturing. You can figure out where the mobile elements are coming from. And so here's an example. We published this a few years ago. This is a wastewater sample. And what you're looking at here is this middle ring. These are the genomes in that sample. This is the proteobacteria from this wastewater sample. And again, we make these mags de novo from the sample. And then out here, these are your plasmids and your AMR genes. So if there's a little tick mark like this one, that means that this antibiotic resistance gene is present inside whatever this bacteria happens to be. Okay, and like it's not limited to 1-to-1. So if you have a row, like if there's a bunch of tick marks along a row, that means that this AMR gene is present in a number of different hosts. Right. So remember, these things jump around mobile elements, right? So antibiotic resistance, one of the reasons why it spreads like wildfire is because it jumps around from organism to organism. So if you treat population with an antibiotic, The resistant organism can pass its AMR genes around through horizontal gene transfer. And this is a way to see it. And so you can also, like, if you have a little stripe this way, that means that whatever this bacteria is, it has all these different plasmids in it. So you can detect multi-drug resistance, right? You can detect promiscuous AMR genes that are present in multiple organisms. So you're simultaneously discovering the organisms and you're discovering the mobile elements that are in them without, again, this is all coming from the same library prep. And so in addition to publishing the AMR stuff, there was a lab in France that published something very similar with viruses. So they said the same idea, except for instead of plasmids, now what we can do is we can annotate the viruses, the phages that are inside the sample, and we can actually figure out who the hosts are of these phages out of the data. And then we pushed it a little bit further. And so the idea here is that imagine you have in your metagenome 2 contigs that are viral. So 2 contigs that have viral genes on them. And they are both touching each other. And they're also both touching the same bacteria. That tells you that they're likely to be part of the same phage, right? So those 2 pieces of information together, they're touching each other and they're touching the same bacteria. They're probably part of the same virus. And so we've shown that to be the case in most cases. We wrote this piece of software called Proximity that we've also integrated in our platform. And so the idea is that we're sort of simultaneously now, what this allows us to do is it allows us to deconvolve viruses. So we can make little mags for viruses, little bins for viruses, and connect them to the bacterial mags that we made, as I described in the last couple of slides. So we're simultaneously, so if you want to read this envio plot, basically this little line here means that there's a bunch of contigs that are touching each other, and they're also inside the same bacterial host. And so that's a vMAG, that's a viral mag, okay? So we get a bunch of these viral mags, and of course we're associating them with their bacterial host at the same time. So here, this is a heat map where basically what you're seeing is these are viruses on the x-axis and bacteria on the y-axis. And so every one of these little tick marks, that's a phage, that's a virus of some sort. We have a genome for it. These are not taxonomic assignments, right? There's actually a FASTA file with the genome of whatever that is. And it's connected to whatever bacteria is over here, right? Again, so we now have a sequence of a virus, a sequence of bacteria, and we know that they're touching each other. Okay? And as you can see, there's tons of them. Now, if any of you have ever tried like isolating a phage, you know how much work that is, right? It's hard. Here we're getting like, I don't know how many that is, 100 phage host affiliations from a single data set. And so it's just another way, again, it's not a brute force method. This is not single cell sequencing. It's just a clever use of information that was originally intended for something else. And we've done a good amount of benchmarking. I'll give you a little bit of an example just to show this is not completely made up. One of the things that we do when we benchmark these is we have long read assemblies. So you say, okay, well, like, how do I know that this is correct? Right? How do I know that your binning solutions are actually meaningful at all? So what we have is in this case, so this is a sample for which we have both a well-assembled long read assembly, right, with nanopore, and kind of a normal sort of short read Illumina assembly. And what we're doing is we're essentially, we're comparing them to see how similar they are. So in this case, we have, this is a fecal sample, we have 73 high quality viral mags that we pulled out, and 25 of them have equivalents in the nanopore assembly, right? ONT is Oxford nanopore. And so we have 25 viruses that are found both in the ONT assembly and the short read assembly. Out of those 25, 19 are perfectly supported. So they're basically the same. And that's kind of like you see this here. In one of these figures, the little triangle in the middle, that's a contig. That's a long read ONT, that's a nanopore contig. And the little circle balls out here, these are the contigs from the long read assembly. So you can see that they're all mapping to the right one. Once in a while, you have either it splits off or something like that. Once in a while, you have what we think are more than one phage in the same host. And so that sometimes makes them bleb into one, like double phage. But in general, the vast majority of these things are in agreement with what we see in long read assembly. And one of the things, I have this asterisk here to remind me to say something. So one of the reasons why this is useful is that it allows you to get these viral mags from short read data. Remember, short read data, like, okay, so this is an ONT assembly, we got 25. And then the short read assembly, there's 3 times more. So short read data is cheaper, and you get way more bins because you get more reads, and so you're able to extract lower abundance things. And so so kind of like one of the nice advantages of this is that you can get all this information out of short read assemblies that you normally can't, and you don't have to dish out all your bucks on long reads. We have we did this is a collaboration. This is a paper that was published also last year from Tan Zhang's lab in Hong Kong. So what they were doing is they were using a different method to try to affiliate viruses with their host, trying to figure out, okay, which phages infect which bacteria. Like, how else would you do it from sequence data alone? One way you can do it is you can look for CRISPR elements. So what you do is you look for CRISPR arrays in a bacterial genome, and then you try to find a virus that has those sequences. And you say, aha, this bacterial genome has CRISPR arrays against this virus. Therefore, that virus probably infects it, right? Because CRISPR is defense against viruses, right? And so one way you try to figure out host range is by doing CRISPR annotation. And so what you're seeing here is they were comparing. Reviewers asked for orthogonal data, as they always do, and so they used our data as sort of the tiebreaker, I guess. And so what you're seeing here is in cases where we have sort of both pairs and both data sets, they're like over 90% in agreement. Okay, so the CRISPR method and our method agrees over 90% of the time. But there's one key difference. So here's a little screen cap out of the paper. In this data set, they were able to find 21 viruses connected to their hosts. And in our data, they found like 2,000. So that's the difference. So when they agree over 90% of the time. But in the proximity ligation data, you have like orders of magnitude more. Okay? So it's a way of extracting tons and tons of these things. Here's some more examples from this preprint we put out. Basically like different kinds of poop, like sheep poop, human poop, cow rumen, wastewater. One of the things that I want to impress on you, so I don't know if you can see it, but like all these little tick marks, those are all viruses and that have hosts, right? That have bacterial hosts. And you know, as I mentioned, like sometimes there's so many, like this guy is so many, you can't even see them. There's about 400 of them in this one. And so, you know, normally it's such a labor, it's so hard to get viruses, like virus host interactions, because you have to culture these things. And this allows you to get hundreds of them from even a single sample. Here's a paper we put out last year with PacBio. Again, we collaborate with them a lot. This is what happens if you like just have a lot of money sitting around, you don't know what to do with, and you want to pour it all into a PacBio sequencer. So this is a sample where they generated an insane amount of PacBio data. One sample, they had 1,200 mags, 400 mags more than 90% completeness from this one sample. 200 mags were lineage resolved. So, right, so because PacBio data nowadays is very accurate, you can actually strain resolve your mags right out of the assembly with the Hi-C stuff. And again, 400 viral hosts and 300 plasmid host associations. And so again, just showing like, if you really want to crank this up, you can start getting these metagenomes that are like insane in how much information they carry. And we can do experiments. So here's a cute little experiment. This is a Timecourse of fecal sample. Day 0 is the last day of antibiotic treatment. So the person was on antibiotics, then they stopped. And so day 0 is the end of antibiotics, and now it's recovery, just regular old life without antibiotics. And what you see with time is mags. So these are genomes. They sort of come up. So this is, your gut getting repopulated by organisms, as you would expect, because you're not getting hit by antibiotics anymore, right? So the flora is coming back. With it come more phages. The viruses are also coming back right here. And at the same time, plasmids and antibiotic resistance genes are going away, right? Which is what you would expect, right? You're recovering from antibiotic treatment. So the AMR stuff's going away. The bacteria are coming back, bringing viruses with them. But this is what you would get in a normal shotgun metagenome. You kind of know the abundance of things and who's there, who's not. But we have an extra layer, right? So we can see not just sort of who's there, who's present, who's not present. We can actually see, now your time course looks like this, right? So here's your day 0, your day 2, day 3. We can actually see which mobile elements are inside of which hosts over time. So we can watch them as they spread through communities, right? We can watch them as they jump from one host to another in some cases. We can watch them as they spread, like increase their host range, etc. And I've been talking about phages, but we're getting the same data for plasmids at the same time. So again, same idea, but now these are plasmids instead of viruses. So we can watch antibiotic resistance, not just kind of sort of in terms of abundance and presence, but actually who is inside of whom. Here's a similar example, except for this is a wastewater. This is a wastewater treatment plant. So you can take samples like, you can imagine all sorts of experiments with this, right? Like I don't have anything meaningful to say about this, just like there's like tons and tons of this information inside wastewater, new bacteria, new plasmids, new viruses. We can watch what they're doing in different stages. What's interesting is actually one of the things we're finding, you can't see that in this figure, but the communities between the three samples are actually extremely different. It's not just like one continuous thing that changes over time. Like if you take these three parts of the wastewater treatment plants, the communities are totally different, which is kind of interesting. And then of course we can kind of go ad nauseum. We can start plotting like phages, bacteria over time, and that gives us metrics of horizontal gene transfer. So in many cases we can watch particular genetic elements move from one organism to another, right, throughout time course. Do this stuff, and what I'll do is I'll show you a little bit of clinical data in fecal transplants. So here's a fecal sample. It's plotted a little bit differently than before. I'll show you what this means. So this is a fecal sample from a person who's in vancomycin or some other antibiotic like that before getting a fecal transplant, right? So you guys probably know, right? If you get a lot of antibiotics, you might develop resistant colitis. And if you do, then the best cure is taking healthy person poop, putting into the sick person, and that magically cures you. Okay. And nobody really knows exactly why. But what we can do is, so we can look at what's going on in these samples. So this is a patient who's been sitting on antibiotics for a while. On the left, this is antibiotic resistance. And so these are the bacteria, the bins. And then these are AMR genes. These are the antibiotic resistance genes. So you can see who's carrying what, right? So there's a bunch of bacteria, they're carrying different AMR genes. This one happens to be E. coli. You can see this E. coli has got tons of AMR genes, and this Klebsiella has tons of resistance genes also, right? Which is kind of like what you would expect in this kind of clinical sample. And on the right, you're seeing viruses and who they infect, right? So you can we're discovering phages that are infecting this stuff. Part of the reason why we're doing it is like we're hoping to get some phage therapy traction, right? So phages are seen as kind of like next generation antibiotics because they essentially survive by preying on bacteria, right? So if, let's say maybe this phage can be used to kill Klebsiella, right? So that's kind of the idea is, can we make better treatments against resistant organisms? But here's what's cool. Here's a cool little observation that we're finding. So this is a sample set from a fecal transplant. This is the donor. This is the healthy donor. You can see they have a nice, robust microbiome. This is the baseline. That's the patient who is about to receive the transplant. You can see their microbiome, this B here, is shot, right? Because they've been on antibiotics, so they have very few organisms. And then they get the transplant from the donor to the patient, and then week 1, this is 1 week, 2 weeks, and 4 weeks after the transplant happens. So you can see in this case, this is what's called a successful transplant, where the kind of healthy, diverse microbiome of the donor seems to have engrafted in the patient, and so they're feeling good, they're feeling healthy. But if you look at the data here, what you will notice is every one of these, 1 week, 2 week, 4 week, they have this yellow stripe, right? They have this stripe of yellow and blue. What does that mean? That means there's an organism inside of these samples that's packed full of resistance genes, right? Doesn't seem to be hurting the patient. The person's fine, but there's a reservoir species in there that's carrying tons and tons of antibiotic resistance genes. And so you can imagine what's going to happen next is after a few weeks, person's going to get recolonized by C. diff, Klebsiella, etc. And they have a ready-made source of resistance genes inside their poop, right? So they can, they can immediately pick it up from these reservoir organisms. And so you can imagine this might be causing recurrence, right? And so wouldn't it be nice if we can detect all these different reservoir organisms and sort of go after them? Because again, right now the idea is let's kill the pathogens, right? Which makes sense, right? The pathogen is what's hurting you. Let's kill that bug. But we're not seeing kind of the pathogen's friends, right? The ones that are maybe not harmful per se alone, but they're acting as reservoirs for resistance genes to give to the pathogens. And then, of course, we're doing this on all sorts of other samples. And so, same thing like pig, sewage. Basically, we have like we do like coral and cheese and like all sorts of microbiome samples. It's really good for discovering kind of like new organisms, etc. And so, so I have a little bit of section after this, just to give you guys a little bit of info about this cool project we're doing now. But this is sort of the pre-summary. So what does this technology do? Remember, originally developed for something completely different. I don't know, maybe you forgot it by now. 3D genome mapping was not mentioned at all, right? What we're capturing is this intracellular touching information, right? And that allows us to vastly improve the recovery of genomes out of microbiome samples, which improves metabolism annotation, allows us to get de novo phages and plasmids sort of binned out of assemblies. It allows us to attribute them to their hosts without culturing, something that's in some cases virtually impossible. And you can do this hundreds and hundreds of them. And you can do AMR profiling without culturing stuff, right? You can watch antibiotic resistance genes in organisms that don't have to pick colonies. Maybe you can't grow them out. And so all this stuff, right? So I represent a company, we sell this stuff, we sell kits, we have this platform that does all this. Almost all the data I've shown you like pops out automatically on our platform. We have it all sort of automated, it's free as part of our various offerings. And so but what I want to do is I want to talk about this project that we just started recently. It was motivated by the following thought. So here's some, I've shown these before, right? You've seen these. Like here's a thing that has hundreds of viruses and their hosts. And if you were to ask like how many phage host affiliations are known in in the sort of the databases, it's usually like in the publication space, it's usually like talked about like as in the hundreds, right? So a few hundred of phage host, like really like unique phage host affiliations out there. And as you can see, we have like more than that in just this slide. Okay? So one of the cool things about this tech is that it allows you to extract something that's very difficult to do by normal microbiology methods. You don't have to pick colonies, you get hundreds and hundreds of these things all at once. And so the opportunity, the idea came and said, hey, what if we generated like a ton of this data? What if we just had some like massive repository of viruses and their bacterial hosts, right? Could we train like AI to learn the rules? Like what causes viruses to choose hosts? What causes plasmids to choose hosts? Why? Like what are the molecular determinants? Like why do these viruses choose that bacteria and these viruses choose this other bacteria? And do it, because the idea is we'll never be able to like pick every colony, isolate every phage, right? Like how do we do this? How do we solve this problem? There's too many of them out there. It's too difficult to do these culturing experiments. Well, maybe we can use big data to kind of come to the rescue. So what if we generated a ton of stuff, a ton of this data and mined it for looking for patterns? You can imagine all sorts of sci-fi kind of potential outcomes, right? Now, I'm in the startup business, so I do a lot of like imagining what is possible, right? But if we had a predictive model, if we could predict a phage, like if we got a bacteria we've never seen before, can we like synthesize a phage to just go right after it? Or vice versa, if we get a phage we've never seen before, can we predict who the host of that phage is likely to be? Would this translate to other viruses? Like would phage rules somehow translate to like RNA virus, like if the next COVID came out or something along those lines? Can we basically start predicting these things ahead of time? Can we use this information to make new phages to combat antibiotic resistance? Like I shown you those fecal transplant experiments. Can we make a virus to say, hey, there's this crazy reservoir organism that nobody's ever seen before. Can we just like kill it so that it stops spreading resistance around, right? And there's all sorts of other things, potentially responding to biothreats, things like that. So we went to the Gates Foundation and we convinced them that this was a good idea. And so they gave us a bunch of money to basically generate a giant repository of phage host interaction data. And so we're doing that now. We're like, this is like, if you're in the space, this is like data just makes you salivate. You're like just like pulling out new clades, new genera of viruses nobody's ever seen. So much of this stuff is coming out. We're sort of like starting to basically redraw, like basically building a new map of phage space because we're getting so many more of them than what's already out there. And then of course, connecting that to big data methods to try to really start doing, sort of new predictive things. And everything I said for phages, we also have for plasmids, we have for antibiotic resistance reservoirs, right? I talk about viruses because that's like the hot thing du jour, right? But again, just copy paste plasmid, copy paste antibiotic resistant reservoirs. Same logic applies. Can we really transform how people view microbiology? We have, another thing we get for free is we have tons of, like we have 11,000 Cas, CRISPR-Cas systems in the data. There's all sorts of other like novel antiviral defense systems in our data as well. Because again, remember what's cool about this tech is that it doesn't require prior knowledge to assemble these genomes, right? Like all it knows is this sequence and this sequence were touching each other, put them together, right? So it doesn't require you to know what this is. You can potentially have a virus that nobody's ever seen, some microbe that nobody's ever seen. And that might carry a completely new defense system that nobody's ever seen before that maybe doesn't look like CRISPR. So there's all this other potential information out there. And so what we're doing is we have this project underway where we're collecting, we're calling it Project Microbiome. This is like every scientist's dream, at least my dream, which is that we are gathering lots of data with no hypothesis to justify. We're just gathering, we're just piling up sweet, interesting data from all sorts of places. A lot of it is wastewater sewage type stuff. We're looking at healthy human poops from around the world just to kind of generate a big old atlas of like, what are all the phages that are infecting humans around the world, right? Disease states, right? Whether it's C. diff, FMTs, or some other diseases, understudied environments, whatever. Imagine coral reefs, cattle rumen, et cetera. Animal poop, of course, is of interest. I said coral a bunch of times. We're doing like caves, like environmental samples, like thermal vents under the ocean, like just anything, like anywhere you can go, find cool, interesting microbes, like we want them. And so we're doing this as collaborations. We're looking for collaborators, and that's like, we talked to Yasna about this at some point. I think that's what got her interested. And we decided to do this with the community, right? So we have this budget to process tons and tons of samples. And we said, let's do it in a way where we give people the data back. There's lots of scientists out there. I bet they would like some free data. And so we're essentially doing this as we collect samples from you guys. We give you back the data. You can keep it, you can publish it, you can do whatever you want. We're using it to aggregate our big old AI database. And so that's underway. So talk to me if you're interested in something like this. I tried to make an acknowledgement slide last night, but I realized I couldn't because it's too much stuff. So I'm just going to show you a picture of us being cute. I also want to give a big shout out to Chun Yu. Chun Yu is actually in this room right now. He was actually an intern with us last summer, and he did a lot of the early like phage AI work that we've been doing. So he didn't make it in the picture, unfortunately, but I pasted it in. So anyway, I'm going to stop here and thank you guys for your attention. I'll take your questions.
So one of the issues we have with microbiome data is depending upon the hypotheses we end up with a dominant organism in the community that we try to remove from the analysis, because we're really trying to look at what effect that organism might be having on everything else, right? Are there technologies that you can envision using the same technology you're talking about, but can you couple that with linkers or other things?
Yeah, I mean, so you can use this with target enrichment. Right, these in the end, these are just Illumina libraries, so you can enrich for specific things. That probably won't solve your problem because you're gonna have to enrich for all the stuff you don't know. I think the best answer I have to that is, and for those of you maybe on Zoom who didn't hear the question was, what happens when you have a very dominant organism that's sucking up all your data and really you're interested in the other stuff? I think the value here is that you will get reference genomes for things that are not the dominant organism, even if they're less abundant. And then your shotgun data will go a long way. Because one nice thing with Illumina sequencing is you get many reads, right? The beauty of Illumina, like even if you don't have the length, you have many reads. So if you suddenly have reference genomes for more bugs that are not your dominant organism, just shotgun data will go further because you'll be able to detect more things that are lower abundance.
Thanks for a great talk. I'm curious if you have or know somebody using this technology who's used it to catalog the fungal component of the human gut microbiome. I know that currently like assembly methods are really limited and it's been a big limiting factor in the field.
Yeah, totally. I mean, I'm very interested. We've done a lot of fungal work. The difficulty with fungal microbiome is that you're almost always dominated by bacteria. So the problem is that the bacteria just wins the numbers game. And so we have some fungal stuff, but like we get really nice fungal microbiomes when the sample is like really dominated by fungus. Like we just did this thing with Rachel Dutton on cheese, like there's got a bunch of molds and stuff like that. The problem is, right, if you have the bacteria tend to outnumber the fungus by a lot. And the other thing is the fungus tends to be much harder to lyse, right? So like sometimes even if we have a pure culture of fungus, like cracking it open enough to get a good library is really hard. So you can imagine in the mixed community, bacteria lyse like nothing relative to the fungus. And so you have two things against you. One is abundance, and the other one is that one will lyse much easier than the other. And so that's the challenge with fungal metagenomes is that it's just hard to get enough fungal sequence out of these samples. Bacteria just swamps everything, but it's possible. I mean, we get them.
So in your various soccer ball diagrams, it did seem that there was some connections between the groupings. Does that have any meaning?
Yeah. So the question is the sort of the signal between clusters, the intercluster signal. So it could have some meaning. Usually the meaning is just that there's reads that mismap, or there are sequences that are shared between different organisms, but there may be just a read that has a SNP in it that lands on the wrong one. So it's to some extent a statistical problem that like reads have error rates and sometimes they'll map to the wrong thing. Sometimes you have sequences that are shared. You know, sometimes there's probably errors, but almost always it's some form of read issue.
What I was wondering is if there's any signal in there from different organisms that are close in space, and so maybe some of them are dead that their DNA is now close enough together that it gets hit by the formulas.
It's possible. The problem is, in order to ligate together, they would have to be able to ligate together.
Yeah.
So it would have to be inside the same compartment or something. Like if there's like a membrane between them.
Well, I was thinking of dead cells that they were growing there and some of the population has died. And
The thing is, if you have a dead cell and it's liberated its DNA, when you do a ligation, what it's going to do is it's going to ligate to itself. Right? Again, like let's say you took a bunch of naked DNA and you mixed it and you threw in ligase, you would expect the overwhelming number of ligations to just be closing up and not really. It's possible. What you're saying is formally possible. I don't think it's
much signal
there's
there.
enough of it to measure. I think the error rates in the reads is like going to be vastly more than that.